Hello, my name is Kierra James, and this is my presentation on cell death. So, what is apoptosis? Basically, apoptosis is cell suicide, but why would a cell kill itself? Well, there are many reasons, but just to name a few, cells kill themselves when they are sick or infected with a virus, so the virus will not spread to another cell. Another example as to why a cell would kill itself is for morphological reasons. So, a fun fact in the womb, babies' hands are webbed together like duck's feet. Cells that web our hands together go through apoptosis, and now we have individual fingers because those cells went through apoptosis. So, what makes apoptosis different from necrosis? Well, necrosis is another type of cell death, but is different from apoptosis because necrosis can sometimes lead to massive tissue injury. Necrosis involves the swelling and rupture of injured cells, while apoptosis is a process. There are steps to dismantling the insides of the cells so neighboring cells do not get damaged. So basically, apoptosis, in simpler terms, is a neater way that a cell kills itself, so everything is still contained, and necrosis is more of a steer way of a cell dying because all of the cells insides get everywhere and it's just not good. So extrinsic and intrinsic pathways. There are two pathways dealing with apoptosis, which are extrinsic and intrinsic pathways. Extrinsic pathways are usually activated by the immune system from outside the cell, and the ligand from that signal binds to the receptor on that cell. So that's what makes it extrinsic, that the signal is coming from the outside of the cell instead of the inside, which was intrinsic pathways. And they are activated by internal damage, like damage of DNA, for example. This is done by the release of cytochrome C from the mitochondria that leads to cell death. So, growth factors. Growth factors are usually a protein or steroid hormone that can stimulate or inhibit cell division. Two types of growth factors are platelet-derived growth factor and epidermal growth factor. Platelet-derived growth factor is a protein that stimulates the development of connective tissue cells and smooth muscle cells. Epidermal, epidermal growth factor is distributed in many tissues and body fluids. Growth factors such as these bind to receptors on the surface of target cells, which leads to kinase activity, leading up to the activation of RAS pathways. So, this figure is the PI3 kinase AKT signaling pathway. So, the pathway shown here leads to the activation of protein kinase AKT. Growth factors that bind to receptor tyrosine kinases activate this pathway. AKT suppresses apoptosis by phosphorylating and inactivating a protein that normally promotes apoptosis called BAD. AKT also prevents cell cycle arrest by activating TOR, leading to cell survival. So, major steps in apoptosis. I will be telling you about that in M. So this diagram shows, kind of sums up the major steps in apoptosis. So when it comes to apoptosis, internal contents of the cell are dismantled. In the beginning, chromosomes condense and the cytoplasm becomes smaller. Then the cell begins to produce something called blebs, which look like bubbles, and they are cytoplasmic extensions. The nucleus begins to fall apart along with the rest of the organelles inside the cell, and then the DNA of the cell is pinched by DNase, and DNase is an enzyme that digests DNA. So eventually the cell is all over the place in small pieces called apoptotic bodies. These apoptotic bodies are then eaten by phagocytic cells. So, what causes apoptosis? So, one well known cell death signal is CD95. CD95 is a protein on the surface of infected cells in the human body. When cells are infected by certain viruses, cytotoxic T lymphocytes is activated and induces the infected cells to initiate apoptosis. Also, apoptosis can be triggered indirectly by the removal of survival factors, and the major site that plays a role in this is mitochondria. So, it is kind of weird that we say mitochondria has a hand in apoptosis since we usually know mitochondria to be major major sources of energy and not really cell death. So, how do mitochondria play a role in cell death? But first, in a healthy cell, there are proteins located in the outer mitochondrial membrane that prevent apoptosis called anti-apoptotic proteins. A protein called BCL-2 is closely related to anti-apoptotic proteins. So anti-apoptotic proteins work by counteracting proteins that want apoptosis to happen, called pro-apoptotic proteins. So basically, the more anti-apoptotic proteins there are present, the less likely a cell will undergo apoptosis and vice versa. Now, mitochondria triggers apoptosis by releasing cytochrome C into the cytosol. This happens when a bunch of pro-apoptotic proteins pile up at the surface of the mitochondria and makes little pathways for cytochrome C to escape into the cytosol. Too much cytochrome C in the cytosol leads to apoptosis in the cell eventually. Another way mitochondria is involved in apoptosis is by when a cell is so damaged it cannot repair itself. So when this happens, the cell undergoes apoptosis by the activity of p53. And p53 binds to a protein called PUMA and inhibits BCL-2, which is, remember, an anti-apoptotic protein. So basically, the p53 pathway eventually activate pro-apoptotic proteins to induce apoptosis within the cell. So, last but not least, this figure is the induction of apoptosis by cell death signals by withdrawal of survival factors. So this figure is showing what exactly happens inside the cell when it receives a cell death signal. So the cell receives a signal that tells it to die. In this diagram, it's the ligand on the surface of a cytotoxic T lymphocyte that binds to the death receptor of the target cell. The binding causes receptors to bunch together and adapter proteins to come to the site, resulting in clustering of initiator procaspase protein that now becomes activated in step 3. The procaspases now activate the executioner caspase called caspase-3. When survivor factors are no longer present, death-promoting proteins accumulate, outweighing proteins that are anti-apoptotic, causing a release of cytochrome C. And cytochrome C forms a complex with other proteins, leading to activation of caspase-9, which is an initiator caspase. Caspase-9 then activates caspase-3, the executioner caspase, causing apoptosis. So this concludes the end of my cell death presentation. Thank you so much for watching.